Okay, so for development of the nervous system, there is a lot of detail in the book of the different stages of development. It's very interesting. I'm not going to require you to know all those, just kind of the basics. Even the PowerPoint is a little bit more detailed than you will need to know. So definitely look at the objectives and know what you need to know. But we're just going to briefly go through it and hit the high points. So really cool thing, from a single fertilized cell you can develop an entire human being. So that's amazing. We start out with a single fertilized cell. But in utero, there are 3 developmental stages. The pre-embryonic stage, which is from conception to day 14, the embryonic stage, which is day 15 to the end of week 8, and the fetal stage, which is from week 8 to birth. So 3 stages. So the pre-embryonic stage, lasting from conception to approximately day 14, the single cell begins to divide as it moves down the uterine tube and into the uterus. During implantation, the inner cell mass develops into the embryonic disc, and it has 2 layers, the ectoderm and the endoderm. This is probably a review from Anatomy and Physiology. I don't know. You guys will have to tell me if it's a review or not. But I don't expect you to know the embryonic stages and everything in detail. So in the embryonic and fetal stages, the embryonic stage is from day 15 to the end of the 8th week. That's when the organs are formed. The ectoderm develops into sensory organs, epidermis, and the nervous system. And the mesoderm develops into the dermis, muscle, skeleton, and excretory and circulatory systems. So the fetal stage is the end of week 8 until birth, and the nervous system develops more fully during the fetal stage, and myelination begins. So the formation of the nervous system actually occurs during the embryonic stage. And it involves 2 phases: neural tube formation days 18 to 26, so that's pretty early on in development, and brain formation begins on day 28. So days 18 to 26, the neural plane forms on the surface of the embryo, extending from the head to the tail region, and it's in contact with the amniotic fluid. By day 26, the tube differentiates into 2 concentric rings, and the inner layer contains cell bodies that are going to become gray matter. The outer layer contains processes of cells whose bodies are located in the inner layer, and that's eventually going to become the white matter. Kind of neat how it all happens. So as the neural tube closes, the adjacent mesoderm divides into somites. The anterior medial part of a somite, which is called the sclerotome, becomes the vertebrae in the skull. And the posterior medial part of the somite, which is called the myotome, becomes skeletal muscle. And the lateral part of the somite, which is the dermatome, becomes the dermis. So it's neat how all our layers are getting formed. Really a sort of fascinating process. So the neural tube, as the cells of the inner layer proliferate in the neural tube, grooves form on each side, separating the tube into ventral and dorsal sections. So the ventral section is the motor plate, and the dorsal section is the association plate. The neural crest separates into 2 columns, and peripheral sensory neurons convey information from sensory receptors to the association plate. You can see where this is leading, right? Or, I don't know, maybe. So it's leading to the formation of the brain, which is pretty cool. It begins on day 28. The hindbrain divides into 2 sections. The lower section becomes the myencephalon, and the upper section becomes the metencephalon. Structures differentiate later to become the medulla, pons, and cerebrum. Pretty neat, right? So the midbrain enlargement retains its name throughout development. The Central canal becomes the central aqueduct in the midbrain, connecting the third and fourth ventricle. So everything's sort of folding and forming these little compartments. It's pretty neat. The posterior region of the forebrain becomes the diencephalon. So that's all the thalamus structures: thalamus, hypothalamus, epithalamus, and subthalamus. The anterior part of the forebrain becomes the telencephalon. So during the fetal stage, you get continued brain development, and the lateral areas of the hemispheres don't grow as much as the other areas. The covered area is called the insula. In a mature brain, if the lateral sulcus is pulled open, you can see the insula. So I hope you guys got to play with some brains in anatomy and physiology, because, you know, that's fun stuff right there. The surfaces of the cerebral and cerebellar hemispheres begin to fold, creating the sulci and the grooves into the surface and the gyri. So we start getting that folding in the brain. And there's some nifty pictures in the book with this. So then the neurons differentiate appropriately after migrating to their final location. And the function of each neuron depends on the area of the brain where the neuron migrates. So processes emerge from the neuron's cell body, and the forward end expands to form a growth cone. When the growth cone contacts the target cell, synaptic vesicles form, and microtubules that formerly ended at the apex of the growth cone project to the presynaptic membrane. So we're building a neural system here, which is, you know, pretty dang cool. So the neural connections sculpt the developing musculature, and the neuronal connections sculpt the developing nervous system. Muscle fiber type is dependent on innervation. Fast twitch muscle is converted to slow twitch muscle if it's innervated by a slow motor neuron. And slow twitch muscle can be converted to fast twitch muscle if it's innervated by a fast motor neuron. So the neural connections actually determine how the musculature is going to develop. So myelination begins in the fourth fetal month, and most of the myelin sheaths are completed by the end of the third year of life. So when you're a brand new little baby, you're not fully myelinated yet. So some of our processes are occurring at different rates in each system. So this brings in to play the idea of growing into deficit. So there might be nervous system damage that occurred early that is not evident until the damaged system becomes functional. So it's like you have these systems, and as the child is developing, systems are starting to come online. You might have some nervous system damage that occurred early that is not seen, is not evident until that damaged system becomes functional, and then you see it. So that growth into deficit is an interesting concept. So you might have some damage to the nervous system that occurred in utero that doesn't develop until the child is 2 or 3 years old, because that myelination is going on until the end of the third year of life. So you might have a neural deficit that doesn't reveal itself until that system becomes functional. So during infancy, there are consequences of sensory deprivation for the infant nervous system. So there are periods that are called critical periods, and sensory deprivation or interruption of development during a critical period can have a huge effect on the development of that system. So there were some studies that were done a while back with monkeys, and I don't really like animal studies, but there's actually a lot revealed with the animal studies where they, during a critical period of visual development, they sewed the eyelids shut on the monkeys, or sewed one of the eyelids shut, and that neuronal system did not develop because it didn't get the appropriate stimulus. So there are changes that are going on that during critical periods that need particular stimulus in order to have the outcome that you want. So, in normal infants and children and adults with extensive cerebral lesions, neck and vestibular reflexes can be elicited by neck movements or by head position changes. So when the head is tilted, information from vestibular gravity receptors is used to right the head by contraction of neck muscles. So in infants, they don't have that yet. That's why you have to support their head when you're holding them. So a lot of our vestibular reflexes have to do with keeping our head level.